Okay, good evening to everybody and good morning for those watching the US, Chandru particularly. It's indeed a great pleasure to welcome you all to the Peninsula Foundation's one of the important events. This is the second in the series that we've commenced, looking at what we call as transformational paradigms, and we commenced with Industrial Revolution 4.0. And as we look at 21st century, the defining moments or the defining characteristics of changes that's going to come about in the 21st century, we all have hear about artificial intelligence, the digital world, and disruptive technologies. But I believe nothing is more important and more significant than the rapid developments that's taking place in the domain of biotechnology and biosciences. And in that context, today's discussion is on the topic of genome editing, its relevance to agriculture and health. And the panelists will look at the implications of policy, safety, and training. We have very distinguished panelists with us, and they are pioneers in the field. And let me start with the leader of the team, my classmate. And it's a great pleasure to have him today with us. And I thank him for agreeing to be on the show today. Professor Srinivasan Chandrasegaran. He's Professor Emeritus in Johns Hopkins School of Public Health. He's been there for almost 2 decades, Chandru.
Yeah,
more than that. Yes, that's right. And so it's particularly a distinct pleasure for me and him. We are classmates from school days, and then and we followed different careers, and he's actually charted out an exceptional trajectory in the field of science and research, and and is a pioneer in the genome editing and genetic research. Professor Chandrasegaran is an alumni of the Presidency College, Chennai. Thereafter, he moved to the US to do his doctorate. He received his PhD in chemistry from Georgetown University. And if I remember correctly, he trained under a Nobel laureate, Ham Smith. Thereafter, he moved for postdoctoral research to Johns Hopkins. And since then, he's stayed there, and he's actually set up his own laboratory there. He's involved himself in the genetic, research, and I've had the distinct pleasure of visiting him and seeing his lab. And of course, Dr. Siva was a student at that point of time. He's written extensively, and when I see his, CV, it goes into a huge number of pages, the kind of papers and contributions that is made in research areas. He's had a distinguished career at Johns Hopkins as a tenured professor till he retired early this year. He's an affiliate member of the Johns Hopkins Academy. In his retirement, he wants to spend more time in India teaching and educating young scientists in research. And that's a topic that we've discussed quite often, that the capacity building in the context of genome editing is an important area that he wants to actually focus on. He serves as the Director of Research at Pondicherry Biotech Private Limited, which he set up along with his brother and is founded by his brother, Gunasegaran. His expertise includes in restriction modification enzymes, designer nucleases, genome editing, synthetic biology, and gene therapy. His lab at JHU pioneered zinc finger nucleases, the first designer nucleus in 1996, and reported the creation of the first designer synthetic eukaryotic, that is yeast chromosome, in 2014. This morning when I opened the paper, I saw the news about Nobel Prize winners on chemistry. And we had 2 ladies being awarded for breakthroughs in gene editing. That is Jennifer Doudna and Emmanuelle Charpentier. And immediately I just thought about Chandru, and he's the pioneer. He's the inventor of genome editing. And well, his time will come, the recognition will come, we are very sure of that. The second person we have today is Professor Rao, who is the professor in the Department of Biochemistry in Indian Institute of Science. He will moderate the discussions today. He is a distinguished, specialist in the field of biochemistry and biotechnology. He obtained his PhD from Indian Institute of Science in 1982. From 1983 to 1989, he's done his postdoctoral research in USA, in the National Institute of Health, thereafter in Switzerland, and also in Cambridge University in UK. He's also published extensively. His research focuses on DNA-protein interactions using restriction modification enzymes and DNA mismatch repair proteins as model systems. The third person we have is Dr. Pankaj Joshi, a very innovative and a great science enthusiast. He's always dreamt of making an impact on human life, and that's what he writes about. With this passion, he's pursued his PhD at G.B. Pant University of Agriculture and Technology in developing transgenic plants that can grow under extreme conditions. One of his key discoveries that he made during his PhD work is the discovery of mitrons in plants during the postdoctoral program at ICGEB, New Delhi. He's spent more than a decade in the industry and in Sigma Aldrich Chemicals Private Limited, now which is absorbed by Merck. He is now the commercial head of director and head of commercial marketing at Merck Life Science India. He also holds an MBA from Olive Business School of Washington University in collaboration with IIT Mumbai. And the fourth person we have on the panelist today is Dr. Siva Prakash Ramalingam, and an exceptional professional. He is with Council of Scientific Industrial Research as a senior scientist. I first met him as the postdoctoral student of Dr. Chandrasegaran in Johns Hopkins when I visited there. He now runs his own laboratory, Siva Prakash Ramalingam Laboratory of Genome Engineering and Stem Cell Laboratory in CSIR. He has a doctorate in biotech from University of Madras, and a postdoc is done with under Chandru in Johns Hopkins. His research focus has been on pluripotent stem cell engineering, and he was also a junior faculty in Johns Hopkins, after which he came back to India and joined CSIR as a senior scientist. Now, let me now hand over to the professionals, and Dr. Rao, you can take the floor, and please go ahead with the program for the evening.
Wonderful. Thank you very much, very nice introduction. I thought before we asked Chandrasegaran, Srinivasan Chandrasegaran to give his talk or views on genome editing. Let me tell you a little bit a personal history between how I came to know Chandra. I first met Chandra in 1993 at a passive meeting on enzymes, on restriction modification enzymes. I had read Chandra's earlier papers on restriction enzymes. Since I also work on restriction enzymes, so I was familiar with the work of Chandra. But in the 1993 meeting, he presented the concept of chimeric nucleases, where he fused two domains from two different proteins and created a hybrid nucleus, which had the potential which he anticipated to be used for genome engineering and things like that. And that was a sensational talk at the passive meeting in Vermont. I distinctly remember the then awarded Nobel laureate Rich Roberts and highlighted this particular talk of Chandra and said that this is the future in the nucleus. So Chandra made a big bang in this meeting, and since then, this is from 1993, I have known him over the last almost 15 to 17 years, where always the paper coming out from Chandra's lab has been absolutely daunting, very, very remarkable, very significant. While I stuck on to the restriction enzyme areas, trying to characterize different restriction enzymes, Chandra actually moved from restriction enzymes and went on to do the synthetic biology, which is the very currently the hot topic in life sciences, and to actually synthesize genomes from the first principles. And that, I think, again, there, he has rewired the whole synthetic the genome of a eukaryotic organism called the yeast, the Saccharomyces cerevisiae, and the landmark paper of in Science that he published has stood out and is always, again, like his hybrid nucleases, stands out and is highly cited paper in the area of synthetic biology. And Xyvo Chandra, being a chemist all the time, studying in BSc chemistry, MSc chemistry, PhD in chemistry, has used simple chemical principles to understand life's most complex processes and has contributed immensely to explore these things. This, yesterday when the Nobel Prize went to Hansen from the Secretary General of the Royal Swedish Academy of Sciences announced about genome editing, I thought Chandra should be one of them. But it's a pity that this has not been included, and I hope, just like Dr. Mandeshwar said, we will get the recognition some way or another soon. There are very nice articles about the CRISPR, the history of CRISPR. Two very nice articles I urge all of you to read is the one by Eric Lander, where he has written in Cells, The Heroes of CRISPR. This was published in 2016. The heroes of CRISPR, in this article, he mentions about the graduate students, the postdocs, who were the first authors of these papers, of Jennifer Doudna or Emmanuelle Charpentier, and others who were never cited at all, and they were never recognized for their work, except as the first authors of the paper. In that, he also mentions, Eric Lander mentions about, famous scientists like John Church, who is also a very deserving scientist in this whole area of CRISPR Cas. John Church has actually mentioned that genome editing nucleases actually started with zinc finger nucleases. And the discovery of zinc finger nucleases, the whole concept of zinc finger nucleases, is Chandra's. And therefore, I think Chandra has done absolutely pioneering work in the area of genome editing, gene editing. Actually, in one of his papers, he's actually used the word genome editing or gene editing for the first time. Nobody else had used these. The other thing I wanted to mention about this whole area about this CRISPR genome editing. Second, back to bacteria. About 60 years ago, Werner Arbor, a Swiss biochemist, discovered he put forward the concept of enzymes that can cut DNA. And this is the whole area of enzymes. And he, again, with two other scientists from Johns Hopkins, got the Nobel Prize for the discovery and the use of restriction enzymes. And now, again, 60 years, 50 years later, 60 years later, it's back to the pages and bacteria where the Nobel Prize is being now recognized for CRISPR, which is the nucleus, and nucleases are just like enzymes, although they're different, non-specific in some sense. So I think if the field has come back to bacteria, I think whatever we learn in bacteria, we learn a lot, and whatever is true for the elephant is true for the bacteria. Or the other way, whatever is true for the bacteria is also true for them. So studying prokaryotic systems, I think is fun, and I think that is where the emphasis is now to be made, especially in a country like ours, where studying prokaryotic research or doing prokaryotic research is much easier. Now, very quickly, I'm sure Chandra himself will tell you about this, but working on this type 2 restriction enzyme, the type 2S called restricted cleavage enzymes. Restriction enzymes are present in all bacteria. You can have one of them, you can have 5 of them. Bacterium like Helicobacter pylori, which I work on, has 44 of them. And the main purpose of restriction enzymes in bacteria is a defense system. So that when a phage or a bacterial virus tries to enter the bacteria doesn't, like the host, doesn't like foreign DNA, and therefore it chops it off. And the chopping is done by these nucleases. And its own genome is protected because the host also produces a partner for the methyltransferases or modification enzyme when genome is modified. So it's a question of unmodified phage DNA entering and modified DNA which is present in the host. It's a question of self and non-self. And this is a very primitive immune system. You and I have very nice immune system, T cells, B cells, antibodies, and things like that. But back in evolution, when life actually started, simple organisms like bacteria and viruses, the RM system of bacteria is a innate immune system. And now the CRISPR that everybody is talking about, the discovery by Doudna and Charpentier, is actually an adaptive. So you have the whole branch of immunology, you have two arms called the innate immunity and the adaptive immunity. And again, these concepts come back again from bacteria that people studied long, long ago. So it's very gratifying that people like us, Chandra, me, Xyvo, of course, have worked on the eminent systems and things like that. And prokaryotic research is again back at the front, and it's very, very. So now I will not tell you more about Chandra's work, but I think he's going to tell us, and it's best to hear it from Chandra. And we had the pleasure in the institute that Chandra spent 9 or 10 months in my lab as a senior Fulbright professor, and I think we benefited so much from his being in the laboratory. The other thing is, during that time, Mark and especially Pankaj, the next speaker after Chandra, very, very nicely supporting us to run a DBT workshop on genome editing. And that was the first kind of a workshop, hands-on experience that was conducted in India for a large number of people coming from across the country. So Chandra actually capitalized this whole thing and ran this workshop. And we hope that even before Chandra comes back to India, that we can still get the support of Mark and others to run this course. The DBT is very, very happy about this course and actually wanted us to do this course of practical hands-on workshop on genome editing in all the 4 corners of India. So thanks to Mark and thanks to Pankaj particularly that this was done. And again, it was of Chandra's. So may I now ask Chandra to tell us about the zinc figure cases and what is done. Chandra?
Yeah, just a minute. I'm sharing my screen.
All right, okay. Is it better that I switch off my video?
No, you can. Yeah, that's fine. Oh, I just want to thank Dr. Madheshwaran and Professor D N Rao for those kind words. I don't know what to say. Thank you very much. And it's great to see all my colleagues, Shiva, Pankaj, and of course, all my classmates. It's a great pleasure. I just want you to take you through the basics of genome editing, okay? It started out when I had my own lab. At that time, I had a simple thought experiment, okay? Genome engineering thought experiment, where the idea here was how to take a mutant gene. You're talking a human cell. It has about 3 billion base pairs, okay? Then you have a small segment of which forms a gene and it's mutated and that causes a disease. How do you take this out and put a normal gene? So when you do that, the hope is it will go back to its normal function. The mutated protein will function normally now. How do you do that in a human cell with 3 billion base pairs? So this is what drove us. You can't use the restriction enzymes because restriction enzymes usually recognize very small sites. And so they will clip the genome too many times and the cells will die. So we needed tools that will recognize a specific sequence in the midst of 3 billion base pairs. Okay, that was our goal. So at that time, we asked, do we have the tools to do this? And it turns out we didn't have the tools. So that's how we got into this research. And if you do this experiment, even if you have the tools, does the can you do it without killing the cell? That was a key question, okay? And if you do these experiments in germline, you'll produce gene-edited animals, okay? Organisms. So we started our work, like Professor Rao said, using this type of enzyme. This is a FOC1, which is a type 2S restriction enzyme. As you can see, it recognizes GGATG, but cuts away from that side, 9-13 base pairs away. The idea here was very simple. We thought this should have two domains, one to recognize this specific sequence GGATG, another one to cleave at this side, okay? So we thought we should be able to separate them, and it turned out we could separate them. So, and they are functional. So this part, FR, is FOC1 recognition domain. This is the FOC1 nuclease domain. So we could separate them and show them that they were active. It's a simple idea, again, like a chemistry problem. If you have water, H2O, right? You take off hydrogen and you put a methyl group, you get methanol. You put a ethyl group, you get ethanol, which everybody drinks, okay? This is the same philosophy. So the whole, what we wanted to do was just replace this part with other DNA binding domains. This way we create an enzyme that will recognize a large recognitions. So the first test for that was shown in the next slide. We took a naturally occurring DNA binding domain, which is a helix-turn helix, linked it to the FOC1 nuclease domain, and created the enzyme, and showed that now this enzyme will cut wherever this new DNA module binds. Okay, so we created a chimeric enzyme. Then we were not satisfied with that. We wanted to make programmable nucleases. So at that time, zinc finger motif was known. This is a anti-parallel beta sheet and with the alpha helix. It's held together by a zinc. That's why it's called zinc finger motif. Now the business end of the molecule is here. These are the amino acids protruding out of the helix from positions -1, 2, 3, 6. They interact with DNA, okay? Normally these 2, 3, and 6 recognize these 3 base pairs. But if you have aspartic acid, it can make a cross-strand contact, okay? So zinc finger motifs, they usually recognize 3 to 4 base pairs, okay? But the beauty of this module is you can link 3 of them together, okay? And they'll recognize a larger sequence. So by adding more and more modules, you can make a big protein which will recognize a very specific sequence, very long sequence, and that will be very unique within the chromosome or within the genome. So then what we did was, this was done by other people. So we took these zinc finger motifs and made a zinc finger protein. Then we fuse it to the FOC1 nuclease domain. As we did in the last slide, this is the nuclease domain. It turns out the nuclease domain of FOC1 has to dimerize, okay, to cleave DNA. So that was turned out to be very lucky for us. Although we generated a 9 to 10 base pair sequence here, because these has to dimerize, and now we have actually 18 base pair sequence. When you do the math, 18 base pair sequence will occur, very rarely within the genome. Okay, so now we had a tool which where we can make designer protein, designer nucleases, and start doing gene targeting experiments. Okay, we tested it. We just took zinc finger protein, fused it with the FOC1 nuclease domain. We just put a linker just to be on the safe side, and we cut lambda chromosome, which is about 48 kb. Okay, it cuts at the unique sites there, right? And we tested another zinc finger fusion that also cut very unique site. So we are very happy. So these things, we have a tool necessary to do the gene targeting experiment. So this is how it works. We published this in 1996. Here we have 4 modules, 4 zinc finger proteins. So that recognizes 12 base pairs. So since this has to dimerize, you can use 2 different zinc fingers. Now this recognizes 12, this recognizes 12. Now you have a 24 base pair recognition. That's very unique within the genome. So you can target and cleave at a specific point within the genome. Around 2010, there came another, scientists discovered another module called TAILS, transcription activator-like effectors. Then they followed the same thing we had done, fused it with the FOC1 nuclease domain, right? And generated talents. These are called talents. The beauty of this, in zinc finger, they recognize 3 base pairs, right? Or 3 to 4 base pairs. But sometimes these zinc finger motifs affect their neighbors. So they are difficult to make. Okay, so you have to go through a lot of selection process. So when they introduced this, it will cost about $25,000 per target. Okay, then talents came. Here they also were similar modules, about 30 to 36 amino acids, but they recognized a single base. Okay, so all you needed was, and the other beauty was they didn't affect the neighbor. They didn't affect the neighbor's recognition. Okay, so all you needed was a simple code. You need 4 different modules. You can fuse them together and make a big protein that will recognize, in this case, 18 base pairs. Because FOC1 has to dimerize, you have 18 here, now 36 base pair recognition. So they will cut at a very unique site within the genome. Okay, so, but these are easy to make comparatively. So now the price has dropped to $5,000 per target. Okay, that was in 2010. Then came CRISPR, Doudna and Charpentier received Nobel Prize yesterday for this work. Here, there is no protein engineering is involved. In the previous slide, you had to engineer these proteins, which is by nature very difficult to do. Okay, this is all in relative terms. This was very difficult. You had to do a lot of selection, optimization. Here, you just much easier. But in this case, CRISPR, you never had to do any protein engineering. You just had a Cas9 protein, but the target recognition is done by RNA. This is what it's shown here. Okay, this is called the synthetic RNA, sgRNA, guide RNA, and this is done by base pairing. And this is very easy to make because you can order it commercially. Okay, when this came out, okay, the technology became accessible to everyone. Anybody could do it. So Cas9 was already there for a long time. You can buy it for $50. This will cost probably another $100. Now you can do the experiment with $150. This shows you the structure of this CRISPR. Okay, just to tell you, what is the principle? All of these reagents do is make a double-strand break within the genome. They are very specific, targeted double-stranded break. Okay, then the cell does everything else. Okay, what it does is cells don't like double-stranded break. They want to, because it will scramble the information. So what they want to do is quickly repair this. So they have evolved machinery to repair this over millions of years, billions of years. Okay, so there are 2 things that can happen here. So there are two repair pathways that are available to ligate this, repair this double-stranded break. One is called non-homologous enjoining. It just seals it up, but that process is error-prone. It makes mistakes. Sometimes it deletes a few days pair, or sometimes it inserts some bases. So it produces a pool of mutants. Here, only the nuclease is given, okay? Let's say you have a mutation here, and you want to correct it. What you do is you use one of these reagents, make a break there, then you can provide wild-type donor DNA, okay? Because now the investigator is providing this. Normally, you have two alleles of a gene. When one is broken, the cell repairs it using the other allele, okay? But in this case, what we do is we trick the cell. We put a lot of this donor DNA. The cell assumes this is the one it's going to use as a template to repair this break. So you can actually correct this mutation, and this is called gene correction by homologous recombination, okay? Also known as homology directed repair. But we can do this, use the same process, but you can insert a gene at a particular locus within the genome. All you have to do is you have to have homology on either side of the break, then you can have anything else in the middle, and that will insert. This is, you can transient addition, okay? So this is the principle. So essentially we use these reagents, and if you want to do just to mutate a gene, you just add only the reagent. If you want to correct it, you add a donor DNA, okay? So these are some examples. So scientists, since these things came out, scientists have edited the mushrooms. You know, when you take a mushroom or potato, if I'm sure some of you would have done it, cut it and leave it out, it will become very black, darken, brown. Okay, then it's not good, you know, not very sumptuous. So what they have done is here, they have knocked out one of the PPO gene, polyphenol oxidase gene. There are 6 isoforms. When they knock out one of those, it reduces the 30% reduction in PPO for polyphenol oxidase, and now they don't brown that, okay? Now, this is another example where they have changed flower color, okay? This is done by Japanese morning glory flower. It's usually violet. What they did was they knocked out a flavonol for diflavonol for reductase enzyme. An enzyme in the pathway that makes this anthocyanin. Okay, when they knock it out, it becomes a white flower. Okay, so these are the things you can do. Then other people have used talents. Here what they did was they edited dairy cattle. Usually cattle have horns, and you know they can hurt the handlers. It can also hurt other cows. So that can produce a big lawsuit. So the way they do previously was they actually pull out the horns. It's a very bloody and painful process. So there was a lot of objection from animal rights groups. So gene editing offered another way to do it. So you can actually target the pole locus, which is responsible for making the horns. Okay, if you delete a small segment of DNA there, then it does not develop horns. This is one cattle. Okay, then here is a interesting stuff where people have targeted a gene, LSDIA1 gene, within pond snails. Okay, usually they are all right-handed. If you look at this snail, the body plan, it goes in the right-handed helix. The shell is a right-hand spiral. When they knock out the gene, this turns it to be a lefty. Okay, but this happens in the progeny. So genome editing, when you do in these cases, you pass it on to future generation. For instance, this cattle has had 6 other cattle. Okay, their progeny, they don't have horns anymore. It was published recently, 2020. Okay. Now, human therapeutics, a lot of people are doing this work because here you can do genome editing, take a person's cells, you correct them and put it back in. So it's like a autologous transplantation, okay, with correct. The idea is once you correct their gene, whatever defect they have, put it back, they should be normal, okay? And that coupled with the stem cell, you can it has a powerful technique. Shiva will talk a little more about that. Then we had Shiva, when he was in my lab, has done sickle cell correction. Now he took one of these iPS cells and corrected it. Normally it's a GTG, which is glutamic acid to valine mutation, which causes sickle cell, and it's corrected here. One allele is corrected to GAG, and this produces 40% of beta globin. It's published there. Okay. Now coming to what is the complexity? Okay, when you have this is in terms of regulatory framework. Okay. There is no regulation when you have natural mutations, okay? And you can have also chemical mutagenesis. It's all accepted, so you don't have to have any regulation. But when you have just SDN1, this is site-directed nuclease alone, without any DNA. Again, that only makes mutations. So again, that can be considered low risk. But when you have SDN2, side-directed nuclease 2, along with small oligonucleotide, this is for gene correction. Again, it can come under low risk, okay? But once you start adding the third targeted insertion of a gene, that has to go to serious, the risk profile increases. So you have to have regulation. So GMO, it's strictly regulated. Living modified organisms, yeah. So this part, SDN, this is site-directed nuclease with a long template, which means inserting genes or traits from other organism or any source, that comes under strict regulation. This is taken out of the draft document of regulatory framework and guidelines. This is for India, okay? And the next, this also gives you an idea. If you just do mutagenesis, like the mushroom, where you knock out a gene, or in the ornamental plant, where you change color, they don't get regulated. They are low risk. US does not regulate them, okay? You can sell them. But the moment you start adding stuff, okay, or doing much more, then you have to have more and more high risk. So it has to go through, in our case, FDA. As far as training, like Professor Rao did, we conducted a workshop. DBT has been very supportive of the workshop. We brought in students from all over India, okay? Just to show you, I am of the opinion, these are two of my nieces. She's born in USA. She came to visit me. I made them, she came during the time of the workshop, made them do the experiment. Okay, this is a genome editing experiment where they have a cell line with a mutated GFP. Okay, so they correct that. So once they correct that, the cell becomes green. Okay, here are Professor Rao's graduate students, Sumedha and Edu. yeah, they helped them to do the experiment. You can see once they did, then when they saw the green cells, their faces light up and they get hooked with science. I think this should be done all the time. I think we should do it, a lot more of this in India. I just wanted to show even high school students. She was a high school student. She was MBBS student, and they can all do these experiments, and we need to train them. And just to finish with the last slide, here are the people who came through the lab and contributed to all the work I've shown you. I'll stop there, and I think Shiva will continue with the medical applications. Okay.
Thank you very much, Chandra.
Yeah.
Very clear, very crisp, and very nice talk. Dr. Mandeshwaran, do we have time for a few questions if somebody wants to ask?
Oh, yes, most welcome. In
Right.
fact, we can tell the audience to raise questions if they want to raise their hand
Yes.
or through the chat mode. They're most welcome to raise questions now
Yes.
on Dr. Chandra's presentation.
Or we can wait after everybody talks.
Anything. If somebody has a pressing question,
Yeah.
don't hesitate to ask. You can type your question also, type the message, and then I can tell them. Well, if there are no questions, then I think we should move on to the next speaker, and that is Shiva. Chandra, will you introduce him? Because,
Remove the.
I'm going to stop sharing the screen to.
Yeah, just a minute.
Yeah.
There is a question. Jeka, would you want to call out, or do you want me to read it out?
Okay.
Oh, we
Okay,
can,
I'll call out.
Yeah.
Firstly, I must compliment Chandra because he's told us many things which was overhead transmission, because we are really not equipped to be able to grasp, I at least am not equipped to be able to grasp the nuances, but certainly I appreciate that it's excellent work. But my question is slightly off the technology part into more from a policy framework. Genome editing now applied to human genetic engineering is rather frightening. I'm reminded of Aldous Huxley's novel, The Brave New World, published, of course, several years ago, where the people ultimately, a class society is created. So this technology in the hands of irresponsible people, how do we manage as a world organization?
So you're talking about eugenics. Yeah, I think in US we have strict policy. You do not work with human embryos. Okay, so the funding, anybody who gets, you cannot use NIH funding for doing any embryo work, human embryo work. Somatic cell therapy, which is taking from an adult cell, let's say he has a disease and there is a gene involved in that defect, you take those cells, correct, like take sickle cell, which is what Shiva will be talking next. That's fine, okay? So that's the rule they have accepted. There was one scientist from China who did do human, embryo using CRISPR, and I think now they have stopped it completely. Even Chinese government took action, and it's completely stopped. And the US is working with the WHO and even British, I think, organizations to control this one. No genome editing of human embryos.
Chandru, I have a question, in the context of what you showed in the changing of colors and the horns being removed from a cattle, etc. When we intervene into the natural mechanisms in plant life or animal life or ultimately in human life. Looking at the plants and the animals, will that have an impact on the biodiversity? Because that seems to be also the constant debate with respect to genetically modified organisms and genetically edited, gene-edited crops issue.
Yeah, in the case of the cows, dairy cattle, a similar mutation has happened 500 million years ago in beef cattle. It's called Angus beef, and they naturally have very small horns. Okay, so it's considered a natural mutation. Mutation occurs all the time. Okay,
that's true.
Yeah, so they're not. They're considered low risk. If the organism is fine, if it grows, like this cattle I showed has given birth to 6 progeny, which is all fine. So that's considered safe because it's happening in the nature all the time. But when you start putting, traits or genes into bring new genes into an organism, then I think the risk, it's a gain of function. So you have to be very careful there, and that has to be strictly regulated. And that's the US policy I just told. Yeah. So the mushrooms, now they can sell it. US has decided not to regulate that.
Okay. Thanks.
Can I ask a question, Matesh?
Sure.
Yes.
Okay.
Please ask.
Yeah, I just want to know what are the commercial, interests and commercial values in this field. That's number one. Number two, in India, we have a lot of, discussion on the BT cotton and the BT brinjal and all that. Why there is so much of a controversy? Could you please explain that?
I think, yeah, there are, with BT cotton, the way they have engineered is you cannot propagate them, right? You have to keep on buying them, and they are very expensive. Second of all, they have not gone through proper regulations, Indian regulations. That's why you have a lot of controversy, especially with the brinjal, right?
Right.
Correct me, Pankaj. Some of you may be familiar than I am.
No, you perfectly pointed out, Professor Chandra, that's the point, because there was a fear also, because they fear onto the horizontal and vertical gene transfer that it may happen. But still, there is a clear guidelines from Genome, right? How you need to plant those BT cotton or brinjals, but it has been a political also, like at some point of time, like people don't want to do that. But
yeah, green
too. But slowly, I would say with these all coming up, there will be a better adoption to the technology, and maybe people start using that.
And people are also worried, Matesh, gene drives, right? Where they want to do mosquito manipulation and release them. Yes, that has potential. So they have to do them everything in a small scale, make sure everything is okay. Then only they can release.
True.
Say something. Hello.
Yeah.
Professor Chandrasegaran, good evening to you. See, I have been into farming for the last 10 years, and all my surrounding areas, the farmers cultivate cotton. Now, I'll tell you the problem. I'll just tell you, because I've been reading on this, and Vandana Shiva, who is a very activist, you must be knowing. Now, the problem with these Monsanto seeds, because now what has happened is the seed market has been captured by these Cargill and Monsanto and these companies. For the developed countries, they have, they go for cases against the farmers, and this transgenic, mutation happens even without the farmer's intention. Means if there is a neighboring field, then this also gets mutated. And for that, then they go for, because they have the, intellectual rights and all that. But here in developing countries, what they have found a way, they have done two things. One is that they call it as a germinating technology, and the other one is terminator. One is verminator and terminator. One is that when the seeds are sown, that time if you do not add certain chemicals, which is again controlled by them and sold by them, and if you don't do that, it will not germinate. And once it germinates, at the time of giving seeds, again they call it as a terminator technology, whereby again the genes are sequenced in such a way that it will not, unless you apply a particular, again, chemical application, external application, which will activate this. And then the seeds cannot be propagated again. So the farmer has to go every time to the market. The practical problem, I'll tell you on ground, we used to have this exchange of seeds between the farmers in the traditional way. Now there is no seeds available except to buy from these people. What used to cost 150 rupees a kg now is costing 1,600 to 2,000 rupees, right? And that is the input cost has. And second thing is BT cotton, Now it was basically meant against this ball moth worms or whatever you call it. Now the predators, they also got, they could not survive because they didn't get these things. Now with the result, what happened? The predator's life is much more, say birds or whatever, insects, spiders, they take longer to revive. And these insects or these whom we are attacking, these worms, they multiply in a hell of a lot of number because there are no predators anymore. Now we keep on applying more and more of these. And that is how the farmers suicide, because the input costs have gone up at least 5 to 10 times. That's why these farmers suicide. So it is actually, if you see, morally and otherwise, and of course, even our scientists here in India, agriculture, they are all supporting all the so-called educated lot. But when you really go into the morality and what is actually happening on ground, this is very dangerous, and it is being again a monopoly being played by all this and the same people. We have, the cancer, so many things are happening, and the farmer has no control. I have no choice now but to get, if I want to cultivate, I have to buy these. This is a problem in a developing country. Yeah, okay. Can I have your views?
We'll come back, yeah. Professor Rao, yeah.
All right. Chandra, you want to continue with this discussion?
No.
Yeah.
We'll go with the talk. Yeah.
Okay. So, okay, we'll come back to this discussion now, Dr.
Yeah, at the end, yeah.
Okay. Okay. Can we now move on? And can I request Shiva to give the talk on genome editing in mammalian human cells?
Okay.
Shiva, you're going to speak for 20 minutes?
Yes, sir. Yes. Okay. Good evening to all. So thank you, Dr. Matheshwaran, Professor Chandra, Professor D N Rao, Dr. Pankaj, for this nice webinar. Professor Chandra has set a stage. So he gave very elegant introduction about how this technology works, about all these 3 gene editing nucleases, from zinc finger nucleases to the recent CRISPR-Cas9. So I am currently focusing on what are the applications, the medical applications of this genome editing in the field. Okay. So these are the most common genetic disorders. So you might have come across, these are the hereditary genetic disorders. So in India, you have very common sickle cell anemia, thalassemia. These are all the hemoglobin disorders, very, very prevalent in Southeast Asia. Okay, and then these 2 disorders affect the blood. So it's called as hematological disorders. And then you have cystic fibrosis, which affects the lungs. And then there are some muscle disorders, which example is muscular dystrophy. And then some of the diseases affects the eyes, ophthalmological diseases. The example is retinitis pigmentosa. And then some of them are neurons, which are affecting the neurons. The example is ataxia. So all these are all the genetic disorders. It's been passed from the parents or the grandparents through the next generations. So the question is whether can we able to fix these. These are all the errors in our genome sequence. Say, so the genome is read in a letter of 4 letters, A, T, G, C, right? So if there is any deletion or base pair substitution or translocation, all these leads to some of these disorders. Okay, so what are the different approaches are currently attempted to treat the above diseases, and why we need the genome editing? So that's the question all of us ask. Okay, so before going into genome editing, I'll just touch upon the gene therapy approach. Last several decades, the gene therapy, gene and cell replacement therapy has been so much talked about and so much been worked on in the field of genetic disorders. Okay, so there are two types of gene therapies. One is in vivo gene therapy. The other one is the ex vivo gene therapy. If you see in the left, you can see the in vivo gene therapy. Okay, so what happens is, say example, if I am giving an example of sickle cell disease, which is a monogenic blood disorder. So in this sickle cell disease, there is a gene called hemoglobin. The hemoglobin gene is very important for carrying oxygen from lungs to other part of the body. Okay, so since there is a mutation in sickle cell patients, there is a mutations, there is a single base pair alterations, there is a substitution. Instead of GAG, the patients, sickle cell patients will have GTG, which alters the amino acid sequence, and then which alters also the blood shape. Normally the RBC, the red blood cells, are round shape so that it can able to pass through our blood vessels easily. So it can take the oxygen from lungs to carry to other parts of our body. Okay. But in the case of sickle cell anemia, because of this single nucleotide alteration, the blood shape gets changed from round shape into sickle shape. So that's why the blood can't able to pass through the blood vessels. So it can able to aggregate and it can't able to pass through the blood vessels. So that's why the oxygen carrying capacity will get hampered. So that's why the sickle cell patients will have different kind of pathology. Okay, so they have leg ulcer, they have anemia. The most common symptom is anemia. So there are different symptoms, array of symptoms they have. Okay, it affects all through the body. Okay, so there is no permanent solution. There is no permanent treatment currently available for the sickle cell anemia. Okay, so what the scientists and clinicians are trying for last several decades is the gene therapy approach. So as I told you, there are two types of gene therapy approach. One is in vivo and the other one is ex vivo. So the gene therapy approach is driven by the viral vectors. The viral vectors are nothing but the vehicles. Okay, so you have a therapeutic gene. In this case of sickle cell anemia, if you take a sickle cell anemia, the therapeutic gene is here a hemoglobin gene. Okay, so the normal hemoglobin gene, the wild type hemoglobin gene without having any mutation. Okay, so you want to put this gene, you want to replace the mutant gene by you putting this therapeutic gene into the patients. But how do you put this therapeutic gene into the patient? You need to have some kind of vehicle to put it into the patient's cells. Say example, here it's a hematopoietic stem cells. Okay, so that's where the viral vectors play a major role. So the viral vectors are like a vehicles. Okay, so if you are coming from home to your office, you use like a bike or bus, car or something like that, some vehicle, right? So something like that here to deliver the therapeutic gene into the cell of interest in our human body, the viral vectors are used. Okay, you take this therapeutic gene, package them into the viral vectors. There are different viral vectors are there. Adeno viral vector, adeno associated viral vector, lentiviral vector, retroviral vectors. So you can take, there are some viral vectors are good, but not all the viral vectors are good. So I will come to that. Okay, so you can package these viral, the therapeutic gene into the viral vectors and intravenously directly inject into the patient cells. Say example, in this sickle cell anemia, you can directly inject into the blood cells, hematopoietic stem progenitor cells. That is called as in vivo approach. Directly you inject through IV approach. The second approach, you can see it in the right side. It's a ex vivo approach. So here also you are using the same therapeutic gene, same viral vectors, but here what you are doing is take out the patient's hematopoietic stem cells and at the laboratory level, you inject these viral vectors into the stem cells at the lab conditions. you deliver it into the hematopoietic stem cells, which is from the patient's own stem cells, and then modify that, the genetically engineered cells, you reinfuse them back into the patient. So the difference between these two things is the right approach is very expensive because you have to take out the cells at the good manufacturing practice. At the laboratory level, you need to inject these viral particles which carries the HBV, the beta globin gene, and then you have to put it back into the patient, the same patient. So it is called as autologous cell replacement therapy. Since it is taken from the same patient and you are putting it back into the same patient, so there won't be any immune rejection here. Okay. So this is very expensive procedure because countries like India, where the developing country, you need to have right infrastructure. The common people can't able to afford it. Okay, it will be very, very expensive. So you need to have some kind of off-the-shelf approach. Okay, so the problem here is, if you see in the bottom, the major problem here is the viral vectors. So the viral vectors, if you deliver it into the cells, it will not go into specific locus. For example, hemoglobin locus at the chromosome 11. It will not go only into the beta globin locus. So it will go randomly in our human genome. It may go into chromosome 10 or it may go into chromosome 1, 2, 3. So it will go and randomly integrate these viral vectors. Okay, it will not go into particular specific locus. Okay, so that is like a, so if you see this, the bottom one, if this is the wheel, so it is not going directly into the mutation site. It will goes randomly in the human genome. So if you consider this wheel as a human genome in the chromosome, so it goes randomly in the human genome. Okay, so we don't want this the random approach. We want only the targeted approach. Okay, so what is the solution for this? Okay, so that's where the genome editing approach comes. Okay, so I am giving you an example for this. So can be able to cure this sickle cell anemia or thalassemia, which is collectively called as beta hemoglobin disorders. Because here in both the diseases, sickle cell anemia or thalassemia, the gene is same, the beta globin gene. Okay, so in sickle cell anemia, as I told, it is a single gene, a single point mutation disorder. It's instead of GAG, it's a GTG. Okay, which affects thousands of people across the world. Okay, so but in the case of thalassemia, the gene is the same, but the mutations are different to different patients. Okay, one patient will have deletion, another patient will have base pair substitution, another patient may have long deletions. Okay, but in the case of sickle cell anemia, across the world from India to Africa or USA, all the patients have the same GAG to GTG mutation. So the question we and others ask is, can we permanently fix this genetic error in human cells so that these patients no need to take any treatment? So they no need to go for blood transfusion, or they no need to take any medicines like tablets, hydroxyurea or L-glutamine or other things. So if you fix that error, the genetic code in the human cells, so those people can be permanently cured, so they will be out of the disease. So that's the question we ask. So before going into the experiments, I'll just give you some background about these two diseases. So as I told you, the hemoglobin is the gene which has the mutation, which alters the shape of the cell, which leads to the sickle cell disease. That's the name it came from. And then in the thalassemia, it's the other side actually. So the mutation affects there are four chains in the hemoglobin gene. Okay, 2 alpha chain and 2 beta chain. Okay, so that's where it forms a protein. Okay, so in the case of thalassemia mutations, so the patients used to produce excess alpha chain and lower the beta chains. So there is a chain imbalance. Okay, so that leads to reduced erythropoiesis. Okay, reduced erythropoiesis means less red blood cell production. So if you see, you might have seen some thalassemia patients, they may be going for regular blood transfusion, right? So why they are going regular blood transfusion is because they have the mutation in this hemoglobin gene that leads to reduced erythropoiesis. Normally, our blood cell half-life is 120 days, okay? But in the case of beta thalassemia patients, it varies from 30 days to 40 days, okay? So the half-life is very less. So they have to produce, keep on producing the red blood cells, but the body can't able to cope up that, okay? So that's why they have to get the red blood cells. So that's why they have to go for blood transfusion, okay? So that is very painful procedure. So you can see it starts from year 1 itself. After the birth itself, you can see the symptoms of these things. Okay, it's a very, very painful procedure. Regular blood transfusion, they have to go for 2 weeks once. So that blood transfusion leads other complications as well. Say example, iron overload, liver enlargement, spleen enlargement, splenomegaly or hepatomegaly. So all these things. So these patients, generally they die within 20 years or 30 years maximum. So some people, it depends on the severity, okay? So it depends on the severity. Some patients, they die very early, but some patients, they die a little later also. So what is the clinical, what is the significant burden globally? So there are around 320,000 children are born with this sickle cell disease every year. You can think about it. It's a very huge health and economic burden for countries like India, Africa, and our neighboring countries. So if you take thalassemia, around 60,000 children are born annually in the world. But if you consider in India, there are around 22,000 children with the sickle cell anemia they born, and 9,000 children with beta thalassemia mutations. So it's a very, very heavy health burden, and also it leads to economic burden as well. So currently there is no cure for these diseases. Only cure currently available is the bone marrow transplantation. Okay, so you might have heard about bone marrow transplantation. It is just you may have a donor if he matches with your human leukocyte antigen, or you can call it as MHC. So you can take, this can be from within your family members like siblings, or you can also get a donor from outside of your family, but that is very, very rare. Okay, so you take the hematopoietic stem cell from the donor, and then you can completely abolish your bone marrow, the patient bone marrow, and then put the donor's hematopoietic stem cells. So that is called as allogenic transplantation. Okay, it's allogenic bone marrow transplantation. So that also has a lot of risk, and it is very expensive. Countries like India, it costs around 18 lakhs to 25 lakhs. Okay, so it is very, very expensive. So common people can't afford it. So it's very expensive procedure. Okay, and only very few hospitals in India, they do these kind of bone marrow transplantations. Okay, so these patients have anemia. That is a very common sickle cell anemia, thalassemia patients. They have common symptom is anemia, and then they have the pain, very severe pain sometimes, and then they go for early death. This is the sickle cell anemia. And for the thalassemia, they have, as I told earlier, they have to go for regular blood transfusion, frequent blood transfusion and hospitalization. So that is very, very painful procedure. Okay, so how to cure disease? Is there a way to cure this mutations permanently? Yes, there is a solution we can use now. Scientists are hoping for using genome editing approach, using zinc finger, talons, or CRISPR-Cas9. You can permanently fix these errors. As Professor Chandra explained in the application slides, you can use any of the one pathway to correct these mutations, either non-homologous enjoining pathway or homology directed repair pathway. Okay, I will come to that. So before going into the genome editing, I will just give you a brief introduction about how this globin gene is developmentally regulated. Okay, so if you see here in the left, this is the normal healthy phenotype. If you have, at the embryonic stage, there is a gene called embryonic hemoglobin is expressed. Okay, so in the embryos, before birth, before even fetal stage, there is an embryo stage, which is where the embryonic hemoglobin is expressed. That is the embryonic hemoglobin which takes the oxygen and passes to the other parts. Okay, and this embryonic hemoglobin is silenced at the first 6 weeks, and then the fetal stage, there is a fetal hemoglobin starts expressing at the fetal stage. Okay, so after that, after a birth, maybe after a couple of months or a couple of weeks, there is a second switch happens. After the birth, this fetal hemoglobin gets suppressed or silenced, and then the adult hemoglobin starts expressing. So this adult hemoglobin starts expressing throughout the life. So this is the case of 99.9% of our humans. So we are all expressing the adult hemoglobin. Okay, but in the case of sickle cell or thalassemia patients, you can see at the embryonic stage, same embryonic hemoglobin is expressed. At the fetal stage, same the fetal hemoglobin is expressed. But in the adult stage, there is no adult hemoglobin is expressed. Okay, because they have the mutation in the adult hemoglobin. So you can start seeing the symptoms after 1 year. Okay, so here you may not able to see the symptoms at the fetal stage or at the embryonic stage, because there is no mutations in the fetal or embryonic hemoglobin. And another thing, the fetal hemoglobin takes care about the oxygen transport from the lungs to the other parts of the body. Okay, but so the question we ask is, can we wake up this fetal hemoglobin gene at the adult stage? So what happens is, there are across the world, there is a tiny population, they express fetal hemoglobin throughout their life. Instead of adult hemoglobin, they express fetal hemoglobin. The switch doesn't happen at all for them. Okay, so the reason is, I will come to that. Okay, so those people are completely normal, even though they have the fetal hemoglobin expression. So there is no clinical abnormality. Okay, so that is because of there are some naturally occurring mutations in the beta globin cluster at chromosome 11. Okay, so there are two types of mutations. One is long deletion in the beta globin gene at the chromosome 11. There are some individuals, even in India, they have these mutations. So those people express only the fetal hemoglobin. Though those people, they don't express the adult hemoglobin. Okay, so those people are healthy. They live normal up to 80, 90 years. Okay, but in some cases, okay, the people, they have sickle cell anemia mutation. Along with that, in addition to that, they also have these long deletions, or there is some small base pair substitution mutations in the promoter region of the fetal hemoglobin. Okay, so that prevents this switch from fetal to adult hemoglobin. Okay, so those people, even though they have the sickle cell mutation, so those people, they express high fetal hemoglobin levels. So that takes care about the oxygen transport from lungs to other part of the body, even though they have the mutation in the adult hemoglobin gene. So their fetal hemoglobin expression is very high in those patients. So those people, they live very normal. They don't show any sickle phenotype at all. Okay, so that's where the question we ask, can we wake up this sleepy gene? Okay, in adult, not 99% of the people, this gene is sleeping condition. It's not expressing. But in sickle cell patients, can be able to activate this gene using genome editing approach. Okay, so that is called so as I told, there are some individuals, very rare, tiny populations, they have the mutations, naturally occurring mutation. Naturally, they have the mutation. Okay, so Professor Chandra told about the thorn, the cow which has which they have the naturally occurring mutation, right? So like that, here also the individuals or sickle cell patients, they have naturally occurring mutation in the promoter region, which is called as hereditary persistence fetal hemoglobin. Okay, so as I told, the expression level of fetal hemoglobin is reduced postnatally below 1%. But there are some individuals, they have the mutations in the upstream promoter region. The promoter is nothing but a vehicle, okay, which drives the gene expression. Okay, so those people, they have the point mutations. If they have the point mutation in hemoglobin F gene, So those people will have high levels of fetal hemoglobin instead of adult hemoglobin. So that condition is called as hereditary persistence fetal hemoglobin. Okay. So the clinical symptoms of sickle cell anemia and thalassemia are alleviated in patients who inherit this hereditary persistent fetal hemoglobin. Okay. So how much you need the fetal hemoglobin percentage? You no need to have 100%. You can just reactivate 20% of the fetal hemoglobin. So that should be sufficient to take care about the clinical crisis, clinical pathology of the sickle cell anemia and thalassemia. Okay. So we were inspired, we and others, there are other scientists were inspired by this nature. Nature has already given a solution. So can we, the question can be asked is, can we create this targeted beneficial mutation in beta globin cluster at chromosome 11 in sickle cell and thalassemia patient hematopoietic stem progenitor cells? That is the cell which produces the blood cells, RBCs, okay, to enhance the fetal hemoglobin levels. Okay, so the advantage here is making enhancing the fetal hemoglobin. It's a one-stop solution. Okay, so why it is a one-stop solution, fetal hemoglobin here? So this is the schematic. So you can see here there are multiple approaches can be used for to fix to correct to cure this disease. So one is, you can see here, you can correct the mutation. You can go, you can take the patient hematopoietic stem progenitor cells, the patient own cells, as I told you earlier, in the bone marrow transplantation or in the Xyvo condition, you take the patient stem cells and then you can use gene editing nuclease, correct the particular mutation. So what happens is it makes a double stranded break. By homology directed repair, you can correct the mutation. That is one approach. Okay, so that may be helpful for sickle cell anemia because you have only one particular mutation across the world. But in the case of thalassemia, each individual, each patient has different mutation. Okay, it is very cumbersome to develop a therapy, cell-based therapy for each and every patient. It will be very cumbersome and it will be very expensive because you need to have each patient to develop a therapy. Okay, so that will be very expensive procedure. Okay, so the second, is there a one-stop solution for this, for both sickle cell as well as thalassemia? So that's where you can create these beneficial mutations and then you can reactivate the fetal hemoglobin. So that can be, that will take care the both sickle cell and thalassemia disease pathology. So you can take out the cells, do the genome editing, reactivate the fetal hemoglobin, and then you can characterize those cells in the Xyvo condition and then put it back into the patients. Okay, so that is what people are trying to do now. We and others we are trying to do. So there is another approach which Professor Chandra also touched, which is called as induced pluripotent stem cells. There is another type of cells which I will talk in my last slide. How we are doing this actually, sickle cell anemia is, so we are trying to create a beneficial mutation. So if you see, this is the left side, this is our hypothesis. So this is the left side, is the disease phenotype. Those people have the mutations, those people have the sickle cell or thalassemia, low hemoglobin level will lead to the disease phenotype. But if you create a beneficial mutation using either zinc finger nuclease, talons or CRISPR-Cas9, you cut this portion and then remove that. That is where the negative regulators are binding here. So you can create using the non-homologous enjoining pathway that will be the beneficial genotype that will lead to the high fetal hemoglobin. So that may eventually lead to mild or disease phenotype. Okay, this is the hypothesis. So how we are doing this? So we have designed, we are using here as a CRISPR-Cas9 approach. So we have designed scissors, molecular scissors to cut this region, the left region and the right region. So we are deleting this 50 kb region in the chromosome 11 at beta globin cluster region. So these are all the sum of the molecular biology experiments. We have created the sgRNAs, we have validated. I am not going into the detail. Okay, so and then we have used the dual sgRNA approach, two sgRNAs. This is left molecular system, here is the right molecular scissor, which exactly cuts, and by non-homologous enjoining, it joins back. Okay, so that leads to very high level of fetal hemoglobin. So this was confirmed by PCR analysis, and then we have confirmed it by sequencing analysis as well. You can see here, this has deleted the expected size expected region in the beta globin locus in human cells. And the next obvious step is we have see these cells, we have done it in hematopoietic stem progenitor cells. So these cells doesn't express the fetal hemoglobin or adult hemoglobin. Okay, so these are the progenitor cells. You need to differentiate them into red blood cells. So in those cells only these genes are expressed. So if you see here in the A figure, so here the left one is the before differentiation, that is the hematopoietic stem progenitor cells, and then we have differentiated at the Xyvo condition. So after differentiation, you can see the pellet, it is like a RBC, red blood cells. Okay, so these cells we have stained whether it is enucleated, because our red blood cells doesn't have the nucleus. So you can see some of the cells are enucleating and some of them already enucleated. And then we have done some of the molecular biology experiments to prove that after making whether these cells can able to reactivate the fetal hemoglobin. Yes, at the RNA level and the protein level, so we have shown that you can able to reactivate the fetal hemoglobin. This is the FACS flow cytometry analysis, which confirms the enhanced expression of the fetal hemoglobin. So further we have done.
Shiva, can you money up now a bit?
Yes, sir. I have just another couple of slides. Yeah. So this is the quantification. We have shown we have achieved almost like 20 to 25%, which is sufficient to reactivate the, which is sufficient to cope up the disease pathology. So this shows that it can be used for patient cells, and it can be it has a scope for the sickle cell anemia and thalassemia patients. So this is the proposed model. So so you can delete the particular region in the chromosome 11, and then this locus controlled region, which forms a loop initially with the HPV gene. After deleting this, it forms a loop with the fetal hemoglobin gene, which enhances the fetal hemoglobin expression. So this is the overall Xyvo approach, how it will be done, or this is almost similar to bone marrow transplantation only. You take patients sickle cell or thalassemia patient blood, that is process is called apheresis, and then isolate the CD34 cells, use the genome editing tools, and then edit the cells, and then you can put it back into the patient. So you can permanently cure those patients eventually. So that is the whole idea of this. So that is the Xyvo approach. But recently, people have shown this will be very expensive, Xyvo approach, as I told earlier. But recently, people have shown in vivo also, you can package these gene editing tools using adeno-associated viral vectors, and then you can intravenously inject these zinc finger nuclease or CRISPR, and then put it into the patient. So this will go into the particular locus, and it will bind, and it will, by homologous recombination, it will attach, and then the particular gene will start expressing, the protein will get produced, and then eventually the patient will get cured. So this is the in vivo approach. You can package these things in a viral vector, and then indirectly intravenously you can inject this. This is the disease, which is Hunter syndrome, which is the lysosomal storage disease, mucopolysaccharidosis, or you can call it as mucopolysaccharidosis. The recently Sangamo Biosciences has used zinc finger nucleases, packaged them, and then they directly delivered. This clinical trial phase 1, phase 2 is going on. So you can go to their website, and then you can see for the more details. So the final slide. So these the gene editing nucleases combined with the induced pluripotent stem cells that got Nobel Prize a couple of years back has more applications in terms of pharmaceutical industries. So they use for small molecule screening, and then these cells can be edited, and then further these iPSCs, induced pluripotent stem cells, can be differentiated into any body cells. So then that can be reinfused into the patients. So there are coupling with pluripotent stem cells, genome editing has huge opportunity. So with this, I thank the funding agencies who helped us doing these experiments. So with this, I will stop here. Thank you. Thank you for the opportunity.
Thanks, Shiva.
Thank
you for your very decent talk. If it's okay with all of you, we take the questions later on. We'll ask Pankaj to give his talk.
Yes,
and then we can ask
Pankaj
questions.
give the talk,
Yeah.
and then we'll take it in the last.
Yeah. Pankaj?
Yeah, perfect. I'm just sharing my screens. I hope my screen is visible.
Yes.
And I'm on. Great. Wow. We started with Professor Rao with giving such a nice overview, and then coming on to Professor Chandra, and always nice to hear from the innovator or pioneer to know about the technology. Wow, what a great introduction of zinc finger nuclease and CRISPR. Like there was a question, somebody said that, oh, which were some kind of overhead projections. So now the things are much clearer, really. They're explained in such a nice manner. Then coming on to the applications where Dr. Shiva explained in very great, I would say great way that how these are critical into the medical application. What I'm going to do is that I'll talk about how this evolution and from the industry perspective has happened and how we have been associated with it. How this genome editing application areas and research in India, who all are researching in India, how big is the market type. So I'll give you an industry perspective. What are drivers, opportunities, and what challenges we are currently facing. Like Professor Chandra did touched one of the important challenge, which was the training part in India, the technology adoption part. And then finally, I'll touch about how Merck is playing a supporting role in India for gene editing community or gene editing technology. So as a Merck is associated with genome editing technology from last 15 years. It started way back in 2005 when we got this Targetron as a gene editing tool for bacteria. So it's quite old when it started with. Then the very important point happened. It was way back when Professor Chandra, who was pioneer in developing zinc finger nuclease, and he came up with the technology which can edit cells. And this was technology, I would say, could be which changed the face, like how a complex problem like sickle cell anemia or HIV/AIDS could be thought of curing them, right? So those are the things which started with just that basic discovery. So Sigma got that, that time it was Sigma. So Sigma got that zinc finger nuclease technology from Sanimo in 2007. And the technology was applied commercially in various ways. So first started with like with zinc finger nuclease, the creation of knockout animals was there, right? Because prior to this, mouse were much easier to manipulate and people used that for the normal experimentation. With this ZFN, a targeted mutation was possible to be done in the rats. The knockout cells or the knock-in cells were easily created with zinc finger nuclease technology, right? So later on, like evolution happened and Professor Chandra told about that CRISPR was like everybody could use it. It was very good, easy to do a homebrew experiment with CRISPR-Cas9. So in 2012, Merck was one of the first company to quickly file a patent onto this part. And I will talk about how patent or IP is very crucial into this technology also. I'll be touching that part. In 2013, we became the first company to bring this technology or products to the market. And later on, we built up the libraries, we built on a new technology like proxy CRISPR, and came up recently in 2016 with a technology called Centennial. So you talk about, it's a powerful combination of tailored biological solution and advanced gene editing technologies to ensure manufacturing process that are both secure and sustainable. So you're making a viral resistant cell line. Later on, recently in 2019, okay, we were studying in whole, maybe a good number of tissue or those all we are. Now there is a capability, like you can study the gene regulation even in a single cell with the barcoded technology that we collaborated with 10x genomics. So you could see how the evolution of the gene editing technology has happened over the period of time from a commercial perspective. How we could talk about, we heard about the medical application, so, but when you look at overall, it's being applied into gene therapies. Dr. Shiva talked about ex vivo, in vivo, and how it is being used very broadly. We touched a bit onto the BT cotton, but it has been much wider use and being used into a lot of agriculture practices where we are working towards creating a cross which can give you higher yields, which can sustain biotic or abiotic pressures. So you're developing those kind of crops. There is a lot of a study into the drug discovery where we are looking for a drug targets or specific phenotypes, and then creating the models for studying the basic sciences. Right, so many things are going on to that part. So when we look at the global market, currently it stands at the 5 billion USD, and by 2025 this is poised to grow at 120% and become a big market of 11 billion USD. And when you look India vis-a-vis to global, we are currently at 0.15 billion USD, and India is poised to become a 0.32 billion USD market by 2025. So the market is also evolving with this all technology coming in. Who is doing what in India, right? Are people publishing a lot of papers into India? Yes. You could see the bubble which are highlighted into map represent they represent the area from where a lot of papers are coming in, and the size of the bubble indicates like how many papers are coming from that area. Like you could see many papers coming from Delhi, close to around 76 paper, and many papers are there from Dr. Shiva's lab. So you could see across the country, whether it's north, west, south, or east, everybody is publishing paper out. In last couple of years, or last few years, there has been 321 publication in India which were related to CRISPR. Right? This doesn't talk about the other things, whether it's zinc finger nuclease or other technologies. So this is papers related to only CRISPR. This shows you the 19% of the research. So 45% of research in our country is still going on to pharma, and 36% of the country's research is still going on to biotech. So you could see close to around 81% of the market belongs to pharma and biotech in the country. So market is quite big. So it's expanding slowly over the period of time. When you look at what India is working on, cell line engineering, genetic engineering, diagnostic application, every one of us would have heard about the kit called Feluda, which came from Dr. Shiva's institute, and Dr. Debajyoti was the person who was working on it. So it's a very economical kit and capable of detecting the COVID-19. And it is being developed using the FN-Cas9 again. So it has got immense diagnostic applications. It has got, when India people are working on, they are working on drug discovery and development. Transgenic plants, I would say if somewhere in the global, the plant research is going on, it's going on very high in India. Many people are working on to the plants in India. And again, the designing becomes very, very tough when you're working onto the plants because not many genomes in plants are annotated. That's another challenge when you talk about these gene editing technologies. Transgenic animals, you'll feel not only about, we heard about cows, in India, a good research is going on to the sheep also. So these are some of the areas where India is focusing onto the genome editing research. So when you're working on these areas, is this IP required or you need some kind of a patenting? Yes. So out-sciencing fields, whether you're working on a basic commercial research, which are being used to develop disease models and tools for a study of disease and gene function, IP rights can come into play. Whether it's into therapeutics, you are working something on CAR-T, HIV, T-cell therapy, yes, there would be need of those IP requirements. Even if it's into industrial bioproduction, where you are manipulating or editing the plant cell genomes, so there also these things comes into play. Other things, whether like you could see that this infectious disease coming very strongly, a lot of research is going on to the diagnostic area. This is again, if people start focusing on that, IP right plays an important role over there. And how we as a supporting it. So currently Merck has 22 CRISPR patents with first in CRISPR integration area. It's about like how CRISPR-Cas9 system, they integrate a new sequence of DNA after cutting that genome. That CRISPR integration is there with us in these countries. Proxy CRISPR, this is an interesting technology with FN-Cas9. We can talk about sometime later about that. And we have paired nicases. You know, one important thing which Professor Chandra talked about, and this is very critical. Today we talk about CRISPR. One thing which matters and which I still favor zinc finger nuclease is toward specificity. Zinc finger nuclease, if you want to cut one codon, it will go still and cut that base, right? But CRISPR being a shorter in length, it can have some off-target effects. Right, but to lower those off-target effects, there has been a development of paired nicases, which have got a very similar look like zinc finger nuclease, and then Cas9 comes and gives a double-stranded break over there. Right, so this again, we have few patents onto that part also. In India, we have filed the patent, and the patents are pending in India. So you could see the lighter one where we have filed the patent, and those are pending. So when you come on to the CRISPR-Cas9, like important point is that, okay, we heard about basic, we know about technology, we know about application, how to use it, what formats it can be used. So, you know, it can be used in different formats, and based on application, you decide the format, right? Whether it's a DNA, RNA, lentiviral, protein, because if you are working on a very hard to transfect cells, you prefer using the lentivirus, right? You heard about the adenovirus when they are using as a carrier. You could use as a protein, because when you're creating a transgenics, it becomes much easier. You know, what happens sometimes, your guide RNA becomes promoter dependent. If you want to do a promoter independent, put in some cells where you don't know whether that promoter expresses guide RNA or not, you could move towards the protein side. So depending your application, people chooses this formats. The different formats are available, can be created, and are available with us also. Now, when we talk about the research, right, we always talk about, okay, what are my growth drivers? While Dr. Shiva was presenting at last, he thanked the funding agencies. So government, when you look at the gene editing part, genome editing part, there has been an improved funding in India towards these genome editing projects. You'll find a specific theme-based project coming in where you can write it and take funding from government. High prevalence of these infectious disease, and you will see nowadays a lot of industry, academia collaborations, and you will see a multiple application across industries. Like we talked about, academia is doing, biotech is doing, pharma is doing. Opportunities, diagnostic is the biggest opportunity nowadays. We talked about personalized medicine. These are the things which are going to do a breakthrough. Population is increasing, food is going down, food security. These are the tools which will make a great impact. We talked about like there could be some bad side of a technology, but good side is much bigger. These are where opportunity lies, and we can take a real role in solving the toughest problem. Challenges, limited expertise, scientific expertise in India, training lab centers. And this is due to these couple of reasons. The technology adoption is very low in India. Still people look for, do you have a reference? Can I reach out to a reference lab? So these are the challenges. How we are serving it. We recently opened a high-end skill development center at IMTech in collaboration with academia. And in the center, in last 6-7 months, despite of lockdown and difficult to reach labs, we started a digital program where we have trained 4,500 plus students in the country on this digital network. And we keep on organizing workshops into the technology like genome editing and training students into India. It's not limited to that. If somebody has an experiment and doesn't have that facility, they can bring samples to our lab, and our trained scientists or the experts can do the experiments over there. It's not limited to genome editing. We do the biomarker experiments over there. So that was the training part. Do we fund some? Merck itself funds from time to time. We have an accelerator program. If somebody plans to open a startup and have a great idea, we have got an accelerator program where they can submit their projects, and if it's good, can be selected. Last year, there were 3 starters from India who got selected for that. Merck offers a financial support for the stimulating scientific projects, and this keeps on coming every year. Core partnership program, we believe on, like if somebody is working on CRISPR, we can have a core partnership and develop and work together. So this is what was funding working, but do we recognize some good works? So in 2019, we started a program called Merck Young Scientist Award, and winner got a good amount of money as an award to support their research and to recognize their good work. So we organized this program alternate years. So last year it was in 2019. This year it will be coming in 2021, because now we are launching it now and will be concluded in 2021, where we honor 12 scientists across the country. This was the one important program that we started. So overall, a summary, my last slide. So currently we are focusing towards skill development, where we are creating an industry exposure. We are helping scholars to develop the right skill sets and adapting this technology advancement. We're looking towards how we can integrate that work and research-based learning for the young researchers, and we can integrate the real-time sample analysis. Like in India, there is a challenge. Not to worry about the lab. Somebody needs the infrastructure. Yes, we have 2 labs, one in Bangalore and one in Chandigarh, who support this kind of a program. So continuously driving innovations in the field of drug discovery, affordable healthcare, fast and accurate diagnostics, and agriculture biotechnology. Thank you.
Thank you very much, Pankaj. Wonderful work, and it's lots of encouraging programs. Thank you very much for the nice talk. We can have a discussion. Participant, is that possible now to have a discussion?
Yes,
based on Shiva's talk and
yes, go ahead.
So feel free to ask questions or write the questions.
Can I say something?
Yes.
Yeah, wonderful talk, Shiva and Pankaj. And I should mention, when I was at Professor Rao's lab, the workshop, Pankaj really went out of the way to give us a lot of stuff. And Merck has been doing a wonderful job in supporting the new technologies. Thanks.
Yeah. Chandra and Dr. Rao, you can start with answering what Putrasen had raised about agriculture and the issues. And we said we'll take it up after all the presentations. So you can actually start
Right.
with.
Chandra, do you want to do that? Because you're the expert.
Oh, I think, yeah, I think some of it came out in Pankaj's lecture, right? You addressed, yeah, the crystal engine. Yes, these people are, it's too expensive for Indian farmers to use these seeds. Okay. But they do give good yields, right?
True.
Yes. So I think they want to use it, but it becomes like Kutarasan said, they also had to buy all the chemicals to go along with it. I think it becomes really expensive. Can we do something about it? I think it should come from the government level, right?
Very true.
Anybody else want to add anything?
I would agree completely with you, Professor Chandra. It should be mutual, right? So industries also have to come forward, absorb some cost onto that part, and government has to definitely subsidize these kind of things, because initially, till the farmer makes the money, the things should be supported onto that part from the cost perspective. But I think he himself answered lot many queries while he was talking about, right? So I believe every new technology has some pros and cons. It's all about how judiciously you use them, how effectively you take it through the government and everyone. So I would say when you are taking up this technology, there should be a strong will from everybody, whether it's farmers, whether it's companies, whether it's government. So everybody have to come together and work strongly in taking this technology further.
Pankaj, I have a question. You did, in one of your slides, you made a point about the technology absorption issue as a challenge and a problem. Could you elaborate on that, please, in the Indian context?
Yes, thanks for asking that question, because that's one of the real problem when it happens to a new technology coming into country like India. Because when government is also funding, or we are also working on that part. We do ask for the references. And same thing happens when we bring something, people ask, oh, did anybody try this CRISPR in India? How many papers has been published in India? So the approach, it's the fear is like that. One, we are with the limited funding initially when you're thinking about some innovative thing. Second, your lab is not trained up to that level. You are perfect. The knowledge is it's immense in India, but maybe you don't have that kind of infrastructure in your lab or do not have that kind of experimental conditions. So that is the one area comes where people look out for reference, people look out for good infrastructure. And this is where we thought to bring up these two labs where we can train people and make them acclimatize to these new technologies. And where we say, you don't worry, you have a thought, you have a great thought process, you know what to do. We will help you get trained in our lab. If you don't have infrastructure, we can bring you to our lab and get the experimentation done. So this is where when we give them a strong support, they are able to work on. I still remember one of the thing when I was interacting with a scientist and he wanted to use a technology. The biggest fear was that what if I fail? I told him, don't worry, if you fail, you don't pay to me. And frankly, they succeeded. They published the paper and they gave even my name onto the acknowledgement. That was one of the very important time I felt, this is one of the way how you can support scientists and give them a strong maybe a small support, they can publish those kind of technologies up.
I think that's a very important point. The fear of failure in science and research. I think failure is a stepping stone to further success, and that is is there a problem in the Indian cultural mindset in that kind of a context, Dr. Rao?
Yes, I think so. To a large extent, I think the failure is always frightening. People don't want to take high risks projects and things like that. We very much do now. Yeah, you're right.
That's right. And Dr. Shiva, I have a question. Beautiful coverage on the sickle disease and the thalassemia.
Thank you.
Do you have the numbers in India as to how many are affected and what is the?
We don't have because we don't keep records in India. That's the problem, right? So, but the numbers are very huge. Actually, it's underestimated. But see, if you see in Chhattisgarh, Madhya Pradesh, Gujarat, even some pockets in Tamil Nadu, especially in Nilgiris, where the tribal populations are there, Kerala, Wayanad region. So it's very, very huge actually. So every year there is an estimate almost like 20,000 children are born with sickle cell anemia. So you can think about if you extrapolated that. So it's a huge number actually. Yeah. So and the people also not diagnosed whether they have the sickle cell disease or not. So those are the things. The diagnosis itself has to come up actually. So.
Which means from the state side or from the government side that the policy intervention is not adequate enough.
Right. So now I think the government is planning, exploring many things. So the DBT, they have a program. Recently, they have a call for proposal also. Set up the diagnostic facility in district hospitals itself, so they can go and do the prenatal screening, all these things. So, but it is still early stage, I think.
So, Shiva, can I ask, how expensive is it going to be to activate the FITA? If it is too expensive, it won't reach the common man, right?
Yes. So the current protocol, it will be very expensive. It will take. It will be very expensive. It will be like 15 lakhs to 20 lakhs. So but the other good thing is some of the state governments like Tamil Nadu or Andhra and Kerala and all. So they have the health insurance scheme. So they fund up to 10 lakhs, 12 lakhs. So maybe if the government is smart enough, they can club these technologies, these cell therapy approaches with these the insurance schemes. Then people can be able to afford, I think.
So how many? The health insurance, is it affordable?
The best example is if I give, so in some of the patients, the state governments has supported the bone marrow transplantation itself. So if you can think about it, so it's almost like the same as bone marrow transplantation, right? So if they can able to fund the bone marrow transplantation, why not the cell-based therapy products? But the individuals can't afford that much money. So
Yeah,
the government
especially the
support has to be needed.
people, right? Poor
Yes.
people.
Yes,
Yeah.
yes.
What about in cancer treatment? How successful can other potential?
Yeah, so we didn't touch upon that. There is a CAR T cells that is going on now. So that seems to be very promising. So there are some products already available, cell-based therapy products based on this CAR T cells. There is a company called Kite, and there are other companies also. There are some products available, but still these are all very expensive. These are all in very early stage, but eventually maybe it will be cheaper, but that will take several years. But countries like India, people, all of them can't afford it. So that's where we need to have something like off the shelf. People are also working on off the shelf. So then you don't need to make for individual patient based things. So if it is off the shelf, then can be used for many patients. So then the cost will come down.
The cost is a big, deterrent for most of the people. And going by, I suppose, if you really analyze it carefully, I think there's a lot of, track record of significant numbers of the population being affected by these hereditary diseases as well.
Yeah.
And Pankaj, from the industry perspective, we have brilliant, companies like Biocon, yourself. And if they all pull together, can't the prices be brought down significantly to enable the government to be able to make policies to address these issues to a large extent on public health?
True, it can be done too, but what happens when it's drug discovery or something comes into the market, a lot of money goes into the R&D. Because one molecule which comes out, there could be millions of molecules for which the company would have failed. So it needs to be a collaborative approach from a government, from industries. They need to subsidize, they need to support them, and then companies can work onto that side and bring those technology into the market. One good thing happened, I would say. Now we are allowed, like companies are allowed as a part of CSR to support research. So if suppose somebody wants to run a project in the country or do a research in the country, and they can apply my CSR part, and if that project goes through the CSR, a good amount of money would be given by the company to them to the research. So I would say this is the one of the recent thing happened in India, which has given a good will give a good bounce to the research fraternity in India.
That's wonderful.
I just want to add up because all these genome editing, the cell-based therapies, without the clinicians' support, we can't do all these things. It is very, very important in India. We need to teach, we need to educate these clinicians.
Yeah.
So they are the people ultimately taking to the patients. We can do research. We can educate the researchers. It is the CRISPR is something like restriction enzymes, as Professor Chandra used to tell. So this is now like restriction enzymes only. Almost every lab in India uses. I think Pankaj knows this. I think so. But eventually, it has to be taken up by the clinicians. So we need to educate them. So eventually, those people only putting these cells into the patients. So that is key in India actually. So some educational part for the clinicians has to be done. So we need to create an awareness about these technologies. There are some hospitals we are collaborating with, say example Fortis and big hospitals. They are aware of these technologies. In fact, they are keep asking when this will be ready, all these things. But those are all very small. A country like India, the population, it's very, very tiny thing. So you need to educate a lot of the clinicians. That is more important. That one thing in India which is very lagging.
Absolutely correct. Capacity building.
Yes.
Dr. Rao, there's a question on the chat box. You want
Yes.
to read it? You can read it.
Yes, I can read it.
Yeah.
This is for Shiva. What was the Swati allegation? I had a general question. So you discussed the pros of gene editing, but what would be the caveat of using it for curing diseases?
So can you repeat it? What disease is this?
No, she said that you had discussed the pros of gene editing, but what would be the caveat of using it for curing diseases? I somehow don't understand.
So is it the
What would be the caveats for curing diseases?
Okay. So the one thing is the off-target cleavage effects. So it may these gene editing nucleases, all these gene editing nucleases, we all three has covered. So it sometimes it may also cut elsewhere in the genome. That is called as off-target effects. So before going into the patients, so we need to make sure thoroughly do the whole genome sequencing. It made double-stranded break only at the indented site, not elsewhere in the genome. So that is very important. So otherwise, it will be like another conventional gene therapy, like using adenoviral or retroviral therapies. So now the technologies are coming up to check how these are, is there any off-target cleavage effects, the whole genome sequencing, circle-seq, all these things. So, but still that those techniques are very expensive. So it will take time. So there may be some off-target cleavage effects. So that we need to take care before going into clinical applications.
Shiva, this is also true that these genome nucleases, editing nucleases, and implementing them good for monogenic diseases. Right? It's easier to do with one gene and all that things like that.
Yeah.
But if you have a multifactorial, then it becomes good.
Yeah. So, sir, is this on multiplexing? Is the question related to multiplexing?
No, about monogenic diseases.
Sorry, sir.
One gene disease.
One gene diseases.
Yeah.
Yes. So, so the success rate is high in single gene disorders, right? So, because in multiple, so it's like more than one gene if it is involved. So it has to correct both the genes.
Yeah.
So the other part is you have autosomal recessive disorder and autosomal dominant disorder also.
Right.
In the case of autosomal dominant, you need to correct both the alleles. So the efficiency has to be very high. So it has to, if you want to correct the mutation, because in primary cells, say example, in the case of hematopoietic stem cells, these are all the quiescent cells. So it can't, you can't culture them very longer period. So those cells, the homologous recombination efficiency is very low. So it happens only in late S and G2 phase. So, but in the case of non-homologous enjoining, it happens throughout the cell cycle. So that's where we are using, we are trying to reactivate the fetal hemoglobin in sickle cell disease. So we are taking non-homologous enjoining pathway rather than going into homology directed repair pathway.
There's another question here. Does that group of human being gets changed automatically, or does the blood group of a human being?
No, the blood group will not get changed. No.
Or is there a chance to get it changed through blood transfusion or bone marrow transfusion?
No.
No.
Yeah.
No, this person had this doubt whether what
I
change
think
of that.
the person is probably wondering about universal donors. Yeah, that's
so the universal donors, yes. Then you need to have the HLA matching. So now there is people are working on it. So you can mutate one of the gene which is called beta-2 microglobin. So then it's a MHC class 2 molecule. So then it can be used for more people in that case. So the CAR T cell therapy also going towards in that aspect, which is called as universal cells.
Yeah, universal donor, that's what they CAR T cell.
Yeah, okay. Both Chandru and Dr. Rao, and of course Pankaj probably would also be involved. I just want to bring you all to a question on the policy with respect to Indian policy on genetic engineering. Do you find it overcautious and overprotective with respect to, encouraging research and applications? That is one. And in the context of Chandru, we've spoken about the capacity building, which Siva also pointed out. Is there a huge vacuum in the context of building capacities in the and awareness in the genetic editing side with respect to our, whether it's education policy or whether training policies or whether it's safety?
if it is up to me, I will start teaching high school seniors and juniors. If they have time during summer, bring them in, let them do genome editing experiments. That's what I would like to do, expose them. That's good because they'll get involved in STEM, science, technology, mathematics, and engineering, right? That's the goal of STEM anyway. Then I don't know, maybe Professor Rao can address this. I think from my point of view, even in US, they seem to be going towards more applied research. If you look at the work we started studying folk 1, that's more basic research. How is the funding for basic research in India? Is it always being pushed towards only applications because you want patents?
I think so.
Some of these take time. And US has invested in basic research for a long time. And I think China is doing a lot now. And I think we are way behind than China in terms of policy and everything.
I think India's problem is people are not enough people are coming into basic science research.
That's one thing. Yeah, that's true. Yeah, you're right. But even, policy-wise, I think, this is all new to the government and to the regulatory agencies. They are a bit fastidious and they're very slow in doing this. And therefore, things have not moved fast. But the last 3-5 years, I think the DPT has taken a very proactive role and is trying to improve. But generally, very conservative. Yes, right. As far as human resources and training is concerned, as Chandra said, I think it's best to start off at high school level. A good exposure and that.
I think that is one of Mateshwaran. It's close to your heart, right? Getting outreach programs to get high school students involved.
We will. Yeah, that is the way I think probably we need to look at it. I fully support your view. I think you need to start early on that and bring in the awareness on science and technology to.
Some plays need to be developed, or some kits which kids can buy it and do the experiment, and so this will bring to people get acclimatized to these things. And another thing, the challenge is that when you look at these all policies, these are quite old. Like these, the zinc finger nuclease or CRISPR is coming in last few years. We have not updated our policies that much. A regular upgradation. And what Professor Chandra pointed out, the basic problem that needs to be addressed. We need to get our kids introduced to these technologies as soon as possible. I love what he did, introducing in his lab to younger kids that gene editing technologies. If they get introduced at that stage, their mind will open up and they will think much beyond a specific point.
That's same as what Shiva was saying, getting clinical people, right?
Yeah, so I think so. That's a point.
Maybe the first year MBBS students should be doing all these things, right?
Yeah.
Yeah, not just going to coursework.
True, and you will find the doctors are the least, I would say they are ignorant about these technologies. If I ask somebody who's MBBS, I talk about a CRISPR-Cas9 or ZFN, they may not be knowing about this technology, does?
You're right.
That's a reality, yes. Jeka, you can call out your question and then probably Dr. Rao can take over.
All right.
The question is, today the RT-PCR tests which are being done for COVID, does it have something to do with genome modification or only multiplication? Because something of that nature takes place, I understand. Is there any relationship in the basics of this?
Yeah, the RT-PCR is just a technique where you take the mRNA from the cell and reverse transcribe into DNA, then you do PCR, right?
Yeah.
It's a multiplication.
Yeah. So you just amplify the signal so you can detect the COVID. That's what. But it's a more definitive test,
Okay.
and it takes longer time to do.
So it's got something to do with DNA multiplication rather than modification.
No, you take the RNA, so
RNA.
somehow the cell's RNA, the virus will have RNA, right?
Yes.
Then you do a reverse transcriptase, which takes the RNA and transcribes into DNA. Then you add a segment sequences, primers from for the viral sequence and do PCR, right? That PCR will, if you have one, it exponentially amplifies. After 30 cycles, you have enough of them to see on a gel or however you want to see it, detect it. Pankaj, please.
Okay.
No, so you answered it very well. So no, both the techniques are very much different. It's just about isolating the mRNA, then converting into cDNA, and then looking at, it's about amplifying that specific area. When you look from a COVID-19 diagnosis purpose, I talked about one of the kit, which was Feluda kit and being innovated in IGIV. So those are the kits which are using these genetic engineering technologies to detect COVID-19. So both the technologies are completely different and doesn't have any kind of a similarity. So you could see when people are creating gene editing technologies in their lab, they may be using the RT-PCR for different purpose or during the experimentation, but both doesn't have a correlation in this. The current RT-PCR doesn't have a correlation to gene editing.
Yeah, maybe I should explain. Cas9, you can knock out the nuclease part. It will bind to the sequence, specific sequence. It won't cut anymore. Then you can use that's how it works, I guess.
Yes.
Yeah.
All right. I think, sorry, anybody wants to say something? I think now it's 8 o'clock now, and I think it's time to wind up. And if nobody has any pressing questions, Madheshwar, can I just summarize? And,
Go ahead with each of you
Yeah.
giving your closure comments. That's right.
Yes. So I think, I'm sure, I hope that all of you have learned something new, especially because of this Nobel Prize that was announced yesterday. And the origin of this work actually is from Chandra's work. I think that's where the exciting part of today's talk is that we had the expert, the pioneer in the genome editing nucleases, speak about his work, and his postdoctoral student Shiva explained the medical applications of it in such a lucid manner. I think looks like curing diseases is a very easy affair. But I'm sure, as all of you said, that doing the basic concept and discovering the basic concept, applying everything is fine by academicians. But I think clinicians should be more involved in it. They should be educated. Probably they are not aware of many of these technologies. And I think the best part is, industries about what Pankaj represents will play a very important role. I think that is the most important thing. And what Pankaj mentioned about work and its capabilities and its willingness to help researchers, help end users, I think that's a fantastic thing. I think this kind of interaction between industry, the academia, and the government, I hope you have enjoyed, and I think all of us enjoyed giving a little bit of talks and all that. But Ishwar, do you want to conclude?
Oh, yes. Yeah. Thank you, Dr. Rao. And I think it's been a fabulous evening. Chandra, would you want to say something before I conclude?
Yeah, I want to thank all the panelists, Professor Rao, of course, Dr. Joshi, and Shiva, and you, Matesh, good to see you. And I'm glad to see all my classmates. It's always nice to see them. And thank you for your kind words. I greatly appreciate it.
Thank you, Chandru. It now remains for me to thank Dr. Chandrasegaran, Dr. D N Rao, Dr. Pankaj Joshi, and Dr. Xyvo Approach Ramalingam for this wonderful evening. It was a great learning experience for all of us. I'm sure the others would have felt as much. but it was also put across very nicely and simply for most of us to understand as well. This is a very, very important area and very, very important subject. It's also the future for the country in terms of food security, in terms of public health, health security. There are many issues that actually will get dominated by biotechnology and genetic engineering.
Yes.
And I think as a pioneer, I think we give Dr. Chandrasegaran a big hand for having initiated this process in the 90s, and he is the pioneer. Thank you so much, Chandru. Thank you so much, Dr. Rao. Thank you so much, Pankaj. Thank you so much, Shiva. On behalf of the Peninsula Foundation, I thank you all for making this evening a wonderful day. Thank you very much.
Thank you. Thank you. Thank you very much.
Thank you.
Thank you all.